innovative idea, which you probably all about how you can keep forest.
Thank you.
So please give me a big clap. Sorry.
No, that's okay. Will that work?
Yes, it will.
Hey everyone, thanks for joining today. Thanks Spencer and Karen for the invite to this awesome event where we are going to offer a little insight into a process that we're coining Mycoregen, which is effectively the utilisation of endemic fungi as a means to phase out woody weeds. I'd like to pay respects to traditional custodians. Oh, thanks.
There you go.
Nice. Yeah, we're good.
Do you want me to click?
Yeah, you click. Thanks. We are coming from Bunjalung country, down in northern New South Wales, and yeah, want to recognise that we are coming onto land here that's not ceded, and really want to make sure we've got full respect for traditional elders and emerging leaders, past, present. So basically, Mycoregen is a modality that is based on biomimicry, which effectively is coming from nature itself. So sharing a bit about my story and how mushrooms came into my realm and how they play a part in this is yeah, just recognising that through organic bush regen and the application of ring barking, I was creating disturbance, and this disturbance was obviously opening the space for endemic organisms in the form of fungi. And witnessing them come through was effectively a revelation for me in the sense of recognising the role they played in the ecosystem, and yeah, getting a chance to kind of get some insight as to how humans can interact with the fungal organisms and use them for purposes such as organic bush restoration.
Speak more into it.
So basically, we've got, let's see, here we are, vegetation, mycoregen. So mycoregen is the act or process of regeneration through the use of mycelium. You can see here we've got a white oyster, Pleurotus ostreatus, coming off of a camphor laurel here. One of the first specimens I kind of had the big green light as far as success, yes, this works. So just talking a bit about how this process of mycoregen actually takes place, we are effectively creating damage that would otherwise occur naturally in the ecosystem through, say, like high winds or a storm, and you've got the bark being broken off a tree, which is the first defense mechanism, and then the chance encounter of fungal spores landing on that substrate and finding a niche where they can effectively take hold and potentially run systemically. So by essentially creating extensive wounds, could you go back one?
Yeah.
Back to the big racing stripe. So that extensive wound up there is kind of showing the ring bark as well as the additional application of the fungal mycelium into that camphor laurel there, and yeah, just stressing the extent of the wounding is really what's going to give that fungal organism, which is currently in that photo in the form of 8mm dowels, inoculated wooden dowels, which will be knocked into those holes you can see that have been drilled, and yeah, basically take hold from there and join together, and ideally run systemically through that woody weed, releasing it directly back into the nutrient cycle and down the trophic levels of decay. So, I mean, this is all very well and good, but I think there is going to come a point here where we're going to have to link this back into Bunya Dieback, and don't worry, it's coming. So yeah, mycoregen supports and speeds up the nutrient cycle, protects and supports the soil microbiome, creates food sources for local fauna. The transpiration cycle remains intact, which is going to aid in the removal or the moving of the water from the ground and up into the canopy until the tree dies. The slow transitioning of the canopy, allowing the understory to flourish, creating food and medicine through mushroom production, obviously chemical free. And a massive one here, which I think is really needing to be stressed once more, is the reconnection to the people, to country. So creating that bridge where we get that active inspiration to go and integrate with the forest that we are tending to, and actually recoup on our efforts and our energy input through the direct harvesting of the mushroom fruit bodies, be it food or medicine. I feel like I'm hogging the microphone here. Rachel hasn't said a word, so welcome Rachel. Thanks for the support through the process.
So I guess now we can talk about mycoregen and Bunya Dieback.
Yeah, exactly.
Yeah.
Yep. So one thing that I really am appreciative of that came from this direction over here was talking about the recognition between the hoop pine and the bunya as the brothers or sisters? Brothers, yep. And so effectively we have got a fungi here, which I'm sure we've got a photo of.
Jump in the slides, you'll jump in the slides.
Jump in slides, sorry. Hold on, give me a moment. Well, basically there's a fungi that has been in the forefront of my attention for some time and has come from interaction with the old growth hoop pine forest stand, and it actually came into play in my life quite some time back, but it took me a while to kind of recognise it and the place it plays in the ecosystem. And really excited to share this one with you. So it's just called Pleurotus tuberagene.
Slow down.
Called Pleurotus tuberagene.
He's very excited. And it's mind-blowing. Like, it's going to be really excited when we get to it.
We're going to get lots of questions, I think.
Yeah,
so
I think so too.
Definitely open for discussion.
So we're talking about the key environments for Phytophthora.
Yep.
We're looking at compacted soil, waterlogged soil. So waterlogged and aerobic soil followed by times of stress. So that could be drought, it could be lots of rain. In high movement areas, poor hygiene practices and land care practices that are not conducive to a healthy ecosystem. And so, now you can talk now.
So with the utilisation of fungi, upon Spencer actually mentioning about the whole Bunya Dieback Symposium, kind of like 3 weeks ago when we were up here for a workshop, we kind of jumped straight back into Paul Stamets' work, and we're looking at mycoremediation and how it could be applicable for the Phytophthora issue. And reading some of his work, we recognised that the work he's done on mycoremediation could very well imply the utilisation of such cultures as Pleurotus ostreatus, Trametes versicolor, and psilocybe species is directly correlated to the management of fungal blights in forests. So over in the US, they've obviously got the sudden oak death, but they've also got other fungi like amarilla mushrooms and other kind of hectic parasitic mushroom that definitely have their place in the ecosystem, but can get a bit out of control, and effectively utilising these fungal strains to effectively outcompete them and fill that niche is kind of the conversation we're wanting to have here today in regards to this Pleurotus tuberagene. So upon finding this fruit body, which I'm sure we've got a photo in the slide.
I do, just...
We're going to have to jump it. Where are we going?
Do you want to talk about it now and we can come back?
Well, yeah, I think that's probably the best option. I'm just going to go straight to it because I've already said it so many times. So
there we go.
Here we go. Pleurotus tuberagene.
We're there.
So a couple of keys here. It's a saprophytic mushroom that grows on, and it's registered online, so to speak, and like numerous citations on it growing on hoop pine biomass. So you can imagine Rachel and I's excitement when we found it growing on bunya 3 weeks ago, and recognising the place it holds and the significance it holds as a culture in the understory of these species or these specimens. And I think really what we're wanting to bring to the table today is the utilisation of this fungal organism as a means to increase biodiversity and increase the soil health. And effectively this mushroom strain, through the use of enzymes, breaks down its food source, but it also can predate on nematodes. So like the enzyme, when the nematode comes in contact with it, effectively paralyzes it and eats it. And I got really poor reception, and it was really late research, but it looks to me like we found a citation on the zoospores, and the fact that the Phytophthora zoospores would appear to be eaten by the enzyme in that same process too. So the potential for microfiltration with this Pleurotus strain is obviously prevalent, along with the Pleurotus ostreatus and the turkey tail and the psilocybe species. So recognising that and the potential for setting up overlays that allow for the water flow to pass through mycelial beds and kind of filter is going to be a potential way to kind of isolate the Phytophthora issue.
And really highlighting that it's an endemic strain of mycelium, so it's a native, naturally occurring, edible strain of mycelium that we found underneath both bunya and hoop pines. And so when we get back to it, talking about land management practices that can promote the development of PTR in the understory of your bunyas to help improve biodiversity in the soil microbiome.
I'm going to jump back to this slide actually, because a big topic there that we've been looking for and wanting to effectively communicate with the Indigenous mob and the use of this fungi is in that second photo there. You can see it's got a large sclerotia attached to the bottom, and the kind of unique thing about this organism is it creates these sclerotia, which are an underground mycelial mass used to overwinter effectively. And this mushroom also occurs in Nigeria, and having scrolled the internet and found that there are citations effectively of it being used as a food and medicine source, recognising the potential massive role it played in the indigenous use as well. Like, how was it used traditionally? Obviously, with the different vegetation that was here and the prevalence of the hoop pine and the bunya, there would have been a large population of them. And really wanting to just bring this back to local mob and put it on the table. So we have got the culture on us today, and
put it on the table.
Quite,
Physically, yeah.
yeah, quite
Actually
literally.
put it on the table.
That's exactly the conversation we're having. There would have been a potential for a damper of the
Oh, yeah.
sclerotia with the bunya, like that would have
And join them
been
together
100%, 100%. So it's a 75% carbohydrate. I think reports of like up to 10% protein, and then a bunch of minerals and other good things in there that are medicinal as well. So yes, jumping around on the slides here a bit.
Where are we going?
I'll go back just because I missed one. So yeah, back to the ecosystem health.
So start here.
What can we do? Right.
So as land care holders, what can we do to help promote the health of our bunyas that we do have existing? So if we've got those paddock trees that we've got in the paddocks as singular specimens, how can we help protect them and try and prevent Phytophthora from infecting them? So do we...
Well, this is effectively where our local expert Spencer is going to be a wealth of knowledge to fall back to. If you've got a bunya that you're concerned of, or a bunya stand you're concerned of more so, because by the looks of it, the Bunya dieback does appear to be more isolated to the individuals and the paddock tree kind of syndrome, which comes back to this here whole consortium issue and the microbiome of the individual specimens themselves and land management practices, like where Rachel touched on it earlier, but if we've got continuous compaction from mowing or continued herbicide use to manage the vegetation beneath, it's nowhere near as a stacked ecosystem whereby it could have been. So in other words, what we're effectively promoting here is the use of naturally occurring vegetation types and replanting adjacent these bunyas to help fulfill the roles in the ecosystem, like the deep layer of mulch that you traditionally find in a rainforest floor.
Yeah. Which you just touched on. So organic land management practices while you're with your bunyas. So ideally avoid mowing the understory, preventing cattle running through, creating these consortiums that can improve the soil microbiome by creating those really complex, diverse ecosystems around that singular bunya. Preventing chemical use, because ongoing chemical use does have detrimental impacts on the soil microbiome. Once you start unbalancing that microbiome, then you can have that risk of Phytophthora taking over. So replace, if we do have weed species in the understory, we can replace those with natives. So looking at the family of the weed species that you have existing and replacing those with natives in the same family. And manual removal of those weed species rather than chemical use. And then mycoregen, which is what we touched on before.
Back to it.
Oh, yeah.
Log stacks are another way you can increase biodiversity in the understory, especially if you're utilising woody weed biomass. So, like for example, if you've got an infestation of camphor or privet, or you've got a tree that perhaps is needed to be coming down near a house for weight reduction, the utilisation of that biomass can actually open up a whole heap of biodiversity through the application of primary decomposers. Like you can see up there, there's turkey tail, Treaties versicolor, coming off some celly wattle. And that was actually a private job that effectively the landowner is now custodian of these mushrooms, and they're placed in such a manner that they are going to obviously break down in their understory. They're not adjacent bunyas, those ones, but the idea there is they're going to be acting like a big battery of nutrients that'll be slow release and providing a whole heap of invertebrate habitat and food and just kind of support that ecosystem.
And releasing spores to continue the production and movement of the mycelium in the system. So in summary.
Cool. Well, so basically just looking at the value of a biodiverse system with all the stratas being stratas being stacked, and just minimising the use of chemical, I'm not going to read because this is ridiculous. Chemical use negatively impacts soil microbiome and allows for pathogens to fill empty niches. We're on mycoremediation with endemic fungi to promote healthy and diverse soil microbiomes and organic land management practices. So ultimately it's just the utilisation of this mycoregen approach does appear to have a huge value in these Bunya dieback instances, where we're going to be able to effectively replenish the microbiome of the forest floor with the fungi that should righteously be there, but due to the fragmentation of colonisation, we've got big holes in the gaps, I guess, big gaps in the whole tapestry of this ecosystem. So just wanting to bring the mushrooms back to knit it all together and stop the isolation of the individual paddock trees.
Yeah. Did we need to touch more a bit on the mycoregen process? Did we need to expand on it a little bit more? You've got a question, go. Do you want me to go back to mycoregen?
Yeah, go back to this.
Yeah.
So how do you actually apply it to the bunya?
Well, in this instance,
you talking about PTR?
Yeah, in this instance, we'll be looking at, there's two direct applications, actually there's multiple applications, but the main way I would go about it would be just getting a mother patch established effectively. So like, I've got jars of mycelium over there, and then you'd effectively add it to either soaked wood chip or straw. It's a really unfussy eater, Pleurotus tuberagene. And once you get that base layer down, you can then add your mycelium or your grain, like the shaken up jar of grain, and then kind of lasagna it up to about 30 centimetres and keep moist, and that mycelium should jump into that substrate, continue to digest it, create the sclerotia, fruit the bodies of the sclerotia.
So in essence, you're utilising grain rather than dough, which you would have seen.
Yep.
Utilising grain. No,
absolutely not. So we're not going to be ring.
Hang on, I can show you. I put this. Yeah, I'll show you a picture. Sorry. We'll go back to PTR. So you can see there there's me holding the PTR that we harvested, and it was actually growing out of one of the limbs on the ground. So in essence, you need to leave the organic matter on the ground as a substrate. So if you were going to try and inoculate the native PTR to that space, you'd be using a grain spawn, which Pierre has, which we can show you, and just running that in the understory. But you've got to leave the biomass in situ because that's its food source.
Well, that's the idea, is to produce a patch, and then once you've got your mother patch, you can then pile your logs on top of that effectively, so that the surface area will allow for the mycelium to jump up into those logs. And then naturally, when it's growing out of logs, it's a really epic experience. To be honest, I hope you all find your own wild Pleurotus tuberagene one day. So it's basically the fruit body comes up out of the wood, and then because it's a white rot that digests the lignin, and the bark of the hoop-pine is cellulose-rich, it remains. It acts almost like an eggshell, and you go to pull the mushroom up, or rip the mushroom up, and it cracked like that, and then out from inside the biomass of the hoop-pine, literally the exact same interior diameter was a sclerotia, like elongated egg shape within the biomass of the wood, and it's just incredible to pull a fruit body, or rather a sclerotia, from a piece of wood and recognise the alchemy that has taken place.
I was actually just going to ask I'll just say mushrooms. So the mushrooms that you're growing out of the bunya that you mentioned that is connected to the bunya in the hoop.
Absolutely.
Are they, and they're edible?
Edible and medicinal, both above the fruit body, as you can see above there. It's like a Pleurotus style fruit body with gills. And when they get old, they do become quite tough, so best eaten when fresh, or if you're going to eat it old, cut it really fine. And then the sclerotia is also edible.
Right, and are they like you said, they were medicinal as well, and are they also hallucinogenic?
No, not at all.
Always got to check.
There's a deeper reason behind that question, which we'll talk about.
Absolutely.
Yeah, I think that's a great idea.
there's an
I
interesting...
got a question.
Okay, I'm coming.
I just wanted to know whether or not if there's none detected in an area already and you're bringing it into an area, could that be a negative?
Yeah, that's a really valid question, and this context is obviously relevant too. We get it a lot with the whole mycoregen process, and I just want to specify it again, but we're using endemic fungi, so they're already naturally occurring in the location. And yeah, obviously this is kind of making the assumption that it's a very valuable piece of the environment and we're bringing it back, which, I mean, based on the fact that we've seen it in intact old growth hoop pine forest and we've also found it growing off bunyas, it looks like the most logical piece of the puzzle to put into that place. But ultimately do hear your concern and recognise there is the potential, obviously, of carrying, like a sclerotia could have come from a bunya that has died from Phytophthora, and effectively there could be mud on that and that could be transported. So yeah, definitely, if you're going to be relocating sclerotia, definitely clean them of material first. Like I would even go so far as to just say don't move sclerotia off bunyas, just to be safe. Like, as we've discussed, it's a pretty hard to notify organism, to recognise it's there until it's kind of taken effect. So yeah, to minimise the unconscious moving of Phytophthora, definitely worth not moving the sclerotia. Does that answer your question?
Yeah. And did we, I'm not sure if we touched on, but within mycoregen, usually what we'll do is we'll go into a space that we're looking at doing the management within, and we'll find existing samples of whatever mycelium we plan on using. So an example is we've got a project here on Sunshine Coast at Changing Habits Farm, and so within that space we use Lentinus sagittal caju, which was naturally occurring within the site, and also Ganoderma affinatum, which was also natively occurring in that site. So the whole plan is that we go and we take cultures from the site, we run them, or Pierre runs them, and then we take them back. So we're not introducing a mycelium that's not already there, and we're using a saprophytic fungi in a parasitic manner. So Pleurotus tuberosum will utilise as a saprophytic fungi, which means it just breaks down organic matter, whereas when we're using a saprophytic fungi...
Dead matter,
saprophytic. Yeah, sorry, so dead and decaying organic matter. So organic matter that's on the forest floor, whereas when we're utilising them in mycoregen, we're damaging the specimen to allow the saprophytic fungi to take over. Does that make sense? Yeah?
Yeah, absolutely.
Cool. Any other questions? Right. Do you want the microphone?
No, I'm all right. Two quick ones. Firstly, you guys, I found this on the internet years ago. They were doing reishi plugs in Camphororals.
Only endemic reishi, yeah.
Yeah, with big scrub. I think, was that you guys?
With big scrub?
For big scrub, doesn't ring a bell.
How long ago was it?
Was it a good presentation or not?
No, it doesn't ring a bell. There's numerous native reishi, Ganoderma species. Yep. Yeah, so we use 2 Ganoderma species for camphor, for example, Ganoderma applanatum and Ganoderma chalceum.
Is that another man?
That's chalceum.
Chalceum, sorry.
Is that a native? Is that the
That's
native?
the, yeah, and that's coming off camphor.
Yeah, that's the one I
There's an interesting paper, and I tried, my internet's not working on my phone here, but it's on Aboriginal use of fungi.
Yeah.
It's a really, but I tried to look it up to find the author, but if you just Google Aboriginal use of fungi, you'll find the paper.
I think where I've come to, having scoured the internet as deeply as I have for PTR information regarding Australian Indigenous use, recognising the best lead is going to be living knowledge, living stories, and wanting to yarn that. Yeah, exactly. Yeah.
Did you have another question?
It's from a land care group application. Would you guys sell kits and things like that?
Yeah, absolutely. Yeah, got all the gears today.
So it would just be a case of following your name?
Yeah, look, I'm already sitting over there at the end of the day if you want to come over and we can have a cross-pollinate and that kind of thing. And I've got dowels, I've got drill bits, wax. We haven't ran through the mycoregen process as thoroughly as we could have, but we also offer workshops. We've got another one coming up, cue the date.
It's in August, but it is part of a bigger workshop. So if we find that we've got enough interest for people to come, we can host another one sooner than that. Ideally, we tend to not run mycoregen in the cooler months just because it is quite challenging to ring bark the specimens, and so when it's cooler, the sap seems to flow a lot slower, and so it's a bit more challenging.
Where do you do those at? Where?
Oh, so it depends on, landholders that would like to offer us their space. So right now we've got a project at Changing Habits Farm, and we did run a campfire, but mainly on broadleaf privet there, which we've run with LSC, so Lenten Sage Caju, because we find that was we found that was a primary weed burden in that space. If there's any landholders that would like to host a workshop that allows us to educate the community on mycoregen, please let us know and we can work something out. Did we have any more questions on mycoregen and the practice, like mycoregen in practice? Yeah. Oh, do you want me to?
Lime as in the tree or lime as in? So depending on the fungal strain, so like I'm growing a fair few endemic strains now in like bucket production, just playing with small scale production, and I've found personally that a lot of the endemic strains don't like a lime soak, which is traditionally what's going to be used to pasteurize your bulk substrate. So I'm now using a cold water soak, just a 24-hour cold water soak, and I've been trialling like Lenten Sage Caju on that, and it's looking pretty good. Like it's jumping off the grain and not being stunted. I tried a bit of rope spawn with LSC that was lime soaked, and it didn't jump at all. So in other words, the lime must have killed it. And similarly, I've seen the same with reishi, like it doesn't tolerate the pH. Certain strains will, but again, it comes back to just trial and error. And generally the cold water soak is going to be the safest bet, especially for such fungi as PTR. But I have actually grown PTR on a lime soak, like Pierre Tuberagene on the lime soak material before, and that was fine as well. So it can hack it. All right. Over there, Rachel.
Huh?
Oh, there's one there too.
Oh, I'll go there and then I'll go there. Sorry.
So just to clarify, you've been able to use this to kill cancers, but have you actually been able to use this with Bunya Dieback yet, or you're just hoping that this fungus that you find will be able to be used in the future?
Look, I actually only found out about Bunya Dieback 3 weeks ago when Spencer mentioned it to me, so I've done some light reading on it. I haven't delved too deeply. I just wanted to put the dots together and bring this to the table effectively and offer that to the community, and then through each individual's eyes, obviously, and that experience of their own Bunya location they might be visiting, the specimens, you guys might recognise a PTR and have that interaction, which can then maybe drip feed the information back, and we can kind of get a bit more of a feel for what the natural stance of Bunya are actually holding fungi-wise. But I definitely see PTR as a big cogwheel, big player in that health of the ecosystem.
Have you done any work with trichoderma?
I'm a professional trichoderma farmer, you could call me that.
Like as a mycorrhizal, would it have any benefits with Phytophthora attack?
Yeah, it's a really interesting question. Definitely trichoderma play a big role in the trophic levels of decay, and as an inoculum, inevitably we're generally trying not to grow it because it's so easily grown in the lab situation. I haven't directly used it other than when I'm emptying my contamination jars, and having recognised Phytophora's role with avocados as well. I've got one avocado that I've grafted successfully that I've planted, and inevitably empty all my contamination onto it, so it's being force-fed large volumes of trichoderma, and currently it looks very healthy.
Yeah, I've heard it working against phyllites.
Yep.
In large mature trees.
Absolutely.
Where fungus will literally fall off the tree.
From the trike?
Yeah, as trichoderma starts entering the system, that attack underground battle goes on, and trichoderma wins and Phytophthora loses.
Yeah, epic. Well, I mean, like in the application of putting myceliated grain out in the form of a PTR patch, inevitably you're going to be inviting that contamination to also grow on that grain, and that'll also, you might not get PTR growing off that one granule of millet, but you might get trichoderma, so that also might play a part. And so I think really what I want to stress here is that one mushroom is not the answer to Phytophthora. It's more so recognising the health of the ecosystem is, and if we can stack these microbiomes to be as complex as possible, perhaps we can leave no space for Phytophthora. So like falling back to a myrtle rust study I read some years ago, it was talking about the propagation of endophytic bacteria on the healthy leaves of a healthy specimen, and the relocation of those leaves into a compost tea and the extrapolation of those cultures then reapplied in the nursery context would allow for these healthy specimens to be, I guess, re-emulated or like propagated, you know what I mean? Like bringing that healthy or cocktail of organisms on like endophytic bacteria effectively that then go out onto the already affected rust trees. It did appear to have some results where they were getting beneficial signs, where they're effectively reintroducing the endophytic bacteria that for whatever reason aren't there and the rust is playing its role. So yeah, just looking at having a beneficial organism in the niche as opposed to a pathogenic or a parasitic.
Yeah, because I wonder if Phytophthora actually is killing all the other fungus in the soil as well. So the soil test coming up with what is still left in the soil after Phytophthora has been through.
Well, that's what Kim and I were having a discussion about previously over lunch. And I think the question was asked about what was the microbial study of an infected soil of a bunya to determine what is existing and the impact that the Phytophthora has, and if we can reintroduce those microbes, or like we're discussing, building that complex ecosystem that can help support the diversity within the soil, which can then prevent the Phytophthora from taking over. But then that's also talking about those organic land management practices that can help support the microbiome rather than destroying it. So the cows, the mowing, the chemical use, all of those.
So what's your perspective on phosphide use?
We were also discussing that.
Yeah, look, I actually haven't looked enough at it to come to some solid conclusion, but my initial fear was the application of it could, especially in spray form, lead to the demise of like the biosphere of the environment that it's being applied on, which could then knock other populations that perhaps aren't currently calculated in the equation of what a healthy ecosystem looks like back, leaving more space open for pathogens. So that's my initial fear. Like, sure, there's short-term results. I haven't looked closely at it to come to a final conclusion, but that's my initial fear.
Is that a question about phosphide?
Yeah.
The peculiar thing about phosphide, my bigger understanding of it is that it isn't a fungicide, and it doesn't kill Phytophthora. It doesn't kill Phytophthora. And it doesn't kill Phytophthora. What it does is boosts the immune system of the plant. I'm not saying that's right, or whether it's doing any damage to the fungi, we need to know that. But there seems to be that sort of, this is like a bit of a magical thing that nobody really knows. It works, so we're not going to look into it, but it does appear to increase the immune system. So the Phytophthora is still present in the soil. So if the Phytophthora is still present in the soil, probably the fungi is still present in the soil.
Excellent.
And what was the thing?
The phosphite, the chemical application is usually being used in this, and it's a type of phosphorus. The scary thing for me would be in WA, low phosphate soils, and you're adding phosphate. That's probably more the concern.
Yeah, and then we can link back to the bioremediation, so utilising endemic, but then we can come back to, where's the stamens page? Utilising endemic strains of mycelium for mycoremediation. So we're adding the extensive amounts of phosphate. We can also look at utilising these endemic strains in the process as well. And so ensuring that we can kind of mitigate, because if we don't know what's happening, then we can always mitigate with mycoremediation.
Yeah, like certain strains will have a certain affinity to clean certain viruses or chemicals up. And Stamets' work is really, we probably should have put a cross-section of an example of it, but yeah, I'll verbally describe it. But yeah, Stamets' work really portrays what chemicals and what mushrooms work hand in hand. I don't remember seeing sulfites on there at all. But,
and superphos.
Anyway, just as the utilisation of different fungi species are going to be applicable for that aspect, and yeah, coming back to the pleurotuber regime is one that I think needs to be looked at more closely in relation with the bunya and the hoop vines.
Any more questions? Do you want to take it?
Thank you.
That's all right.
Thanks. I don't know microbes or fungi at all, but just from the Western Australia, and if adding phosphorus to the soil helps those plants, isn't it? I mean, it's the mycorrhizal fungi who add the phosphorus we would give the phosphorus to the plants in exchange for the sugars. But if the plants aren't getting the phosphorus from phosphite, then it doesn't give the sugar anymore to the mycorrhizal. Mycorrhizal, so the mycorrhizal drops off.
100%.
So in the fact that phosphite works in the first place might suggest, like you're saying, that the mycorrhizal fungi or the whole biome itself is impaired.
Exactly. Yeah, it's looking at the bigger picture and recognising it's not just the one part of the equation. I mean, sure, it's a short-term fix, but I think we might see longer term repercussions.
The whole ecosystem. Any more questions? All right. Thank you very much for having us today.
Thank you.
Our website's currently under production, so we're just updating it. But if you'd like to follow us on We Roast Earth, you can kind of on Instagram. You can kind of see what we're doing, see the mushrooms that we're growing and the projects that we're working on. Thank you.
And as mentioned, if you're wanting to get any mycelium or have to know more about the mycoregen process, be over there in the corner.
Got a video coming out soon as I can copy.
Thank you.
Oh, yeah, be on our website.
There will be, when we do websites, I'll talk about on Instagram. We've got